Hi, today's video is going to be walking you through how to create IGV plots or Integrative Genomics Viewer plots in Pluto for epigenetics experiments. Epigenetics experiments, like the ChIP-seq experiment that I'm looking at right now, it's common to want to visualize the called peaks or consensus peaks, like one shown here, on these tracks on a per sample basis. So in this experiment, I have an input sample, I have a sham and a stroke condition, and these were mouse samples, and again, a ChIP-seq experiment. So here you can see the IGV plot that I've created in Pluto. We're going to break this process down step by step for creating a new one. You can see I've already performed a couple of other analyses on this ChIP-seq experiment. We'll create a new analysis to start a peak analysis from scratch. I'll choose the peak analysis option here from my no-code analysis catalog. And in this area, you can see I can read about this kind of analysis and what I can do, and also access additional help resources. I'll create a new analysis to get started and set up my analysis parameters here on the left. So you can see that this is really pretty simple and straightforward. I can choose to include all of my samples in the IGV analysis, and I only have 3 here, so that's what I'm going to do. But if I wanted to, I could choose to select only specific groups of samples. I could do that grouping by antibody, for example, so if I only wanted to include my target antibody, I could do that. I can also filter down by antibody and my stress condition that you can see in my sample data table, and then really specify if I just wanted the input versus the sham or the input versus stroke, and so on. Running this analysis with all samples will create an IGV plot that I'll then have the ability to customize. The IGV plot shows by default all of chromosome 1, so that's what we're looking at here, are all of the peaks that were called in the sham sample, all of the peaks that were called in the stroke sample, and then our original input sample, which is what's used for calling consensus peaks in these 2 target samples. We also see a track here for the called peaks, which we'll be referencing for analysis. So I can head over to my plot tab to view the different customization options that I have for this plot. So for one, I like to order and put my input samples in the beginning, and then I'd also like to color my samples not only by the antibody, which is how they're colored by default right now, but also including that stress condition, so if this was a sham or stroke. So I'll add stress as a variable to color by here, order my legend appropriately, maybe make this input 1 gray, make the sham a low color, and then the stroke a deeper color here. So now we've got a preview of what our new legend will look like, and I'll click Save Changes to see that on the plot. Okay, so now our samples are in the order that I've specified here. I can also edit the legend labels themselves. So this input is showing NA right now because it has an NA value here in our stress column. I'd like to clean that up, so I can just delete that out of the name and call this input. Click Save again. Now getting into a little more specifics on the science, what we'd really like to do here is not just look at a single chromosome like chromosome 1, but typically look at a particular locus of interest. So in this case, I know that I'm interested in a gene called BTF3, so I can type that into the search menu here and click Enter. And the IGV plot will update to show me this gene, BTF3. You can see here the ref-seq track at the bottom, along with any called peaks that were called in the BTF3 gene or nearest to the BTF3 gene. So I can clearly see here that the stroke condition had higher binding around this BTF3 locus than the sham sample. And then I have my input sample here where you can see very low background noise, which is a good thing. To save the IGV plot at this view, I can now say Set to Current View. You'll see this updates with the locus that I'm looking at right now. I can always change that. So if I did want this a little bit closer to my sample labels, I could have that here, but I like it so that I can see this ref-seq gene in this view also. So I'll go ahead and set that current view and then click Save to update. Great, so now we have this IGV plot looking the way that we want. I'll close out of this so we can view it on the experiment itself and answer a few more questions. So I've mentioned consensus peak calling and a couple other aspects of the methods. You can view the methods section for this IGV plot in this More menu and take a look at how those peaks were colored. In this case, we colored by both antibody and stress. These method sections are written that they can be copied and pasted directly into your publication. The method section also mentions bigWig files, which you can find under the results. So here you can see or download the bigWig file for each of our target samples, the input sample, and a bed file for the called peaks track is also available. That's how to create an IGV plot in Pluto, along with view its methods and results. The final step that we'd really like to take a look at is exporting this plot for publication. When I'm ready to finalize this plot for publication, I can come in here to our export experience, resize this, and really get it looking the way that I want. Even drag this plot within this view to make sure that that BTF called peak that I'm wanting to call attention to is aligned correctly. I can move my legend around, such as putting it down here at the bottom, and maybe even making this horizontally aligned instead so that I can have it centered, let's say right there. When I'm happy with how this IGV plot looks, I can download it in a variety of different formats. That was creating an IGV analysis in Pluto, which is one of many analyses available for epigenetics experiments such as ChIP-seq, CUT&RUN, or ATAC-seq. Thanks for watching.